Hello, everybody. Welcome to the afternoon session. Sorry for the delay. That was on purpose, just to give some time for people to wake up from siesta. And let me start introducing you. You can move on. Fernando Muro. This is Spaniard, but he's coming from University of Gutenberg in Mainz. And he's going to talk to us about the paper that I read in PNAS a few months ago, and I was profoundly impressed by it. So we invited him as the first author to tell us the story of this transition that you will learn.
Hello. So, first of all, many thanks for your kind introduction and for inviting me and giving me the opportunity to talk about our work here. So the title is The Way to Complexity, Rethinking a Major Evolutionary Transition. So this is about a major evolutionary transition, the origin of the eukarya. Okay? It's going to be a multidisciplinary work where you will see that it combines many fields. Computer science. I'm a computational biologist training, right? But also I have experience in physics and artificial intelligence, neural network, and the team is very heterogeneous. So the major evolutionary transition is the origin of the eukarya cell. As you know, it's one of the most important events in the history of life on Earth. Right? It's a biological major transition in the sense that it is in between a simple world confined in simple unicellular cells, archaeas and bacteria, and a much more complex world grounded in eukarya cells. We are eukarya, and our cells are eukarya. From all the major transitions, together with the origin of life, it's one of the most important. Are the most important. Why? Because they unlock the pathway to subsequent transitions: sexuality, multicellularity, sociability, and so on. Without the emergence of eukarya, life in our planet won't be as we contemplate it today. So 60 years of research, and the research is grounded mostly in the structure of the cell, fossil records. So working with archaeas that, as you know, are extremophiles, so very difficult to grasp, very difficult to grow, to cultivate. Genomes that are saturated after 2 billion years of saturation. And we come with a completely different approach, a quantitative way. And we would like to answer the very same question that people is trying to answer for many years, is how this happened. But I hope that at the end of the talk, you will see that we are opening the gate to a new question: is why this happened. It's widely accepted that the endosymbiotic theory explains the origin of eukarya, right? Basically, we have our prokarya world, archaeas and bacteria, cell A and cell B. One engulfed the other one, and endosymbiosis gave rise to a world much more complex, grounded in the cell E, eukarya cells. The increasing complexity was not only because, you see, this is the alpha protobacteria that become the mitochondria. The size is 500 times bigger. So the increasing complexity is not because of the size, or because we have a much more complex structure of the cell. We have now DNA confined in a nucleus that is membrane bound. Not only the DNA, the spliceosome is contained within it, separating transcription from translation. We have 14 different organelles, and as I was saying, the alpha protobacteria become the mitochondria. The complexity arises from the interaction of all these elements. And just think that within a muscle human cell, there can be up to 2,000 mitochondria. So the interaction between all these elements define the complexity, or the complexity that arises from that. Regarding evolutionary time, it's pretty much unknown. It's a review from Nature last year, 24, yes. And it's something in between 1.8 and 2.7 billion years ago. But 1.8 is for sure, because we have fossil evidence that is validated by the scientific community. We have also fossil records for 2.4, but still is not validated. In between, a lack of knowledge. So there is a lack of phylogenetic intermediates. And this is referred as the black hole in the heart of biology by Nick Lane and others. And here we come with our approach, right? It's a quantitative way, and instead of working in the structure of the cell and the same ideas, we work it in a quantitative way. And what we decided is to study how the genomic transformation was carried out in order to allow such an increase in complexity. We are going to study. I remember we were discussing about that, and I said no. We come back to the grounds of molecular biology. Let's start by the ones we know the more, because we have the three-dimensional structure of the chromatin. We have RNA. So how do we study, especially this genomic transformation, especially in the sense of gene regulation? And then we decide clearly, let's go start and let's go to study something like an atom of information. That will be the protein coding gene, right? And the protein that is coding within the protein coding gene. As you know, protein coding gene is the chunk of DNA that is coding for the protein. From all the different possibilities to study here, the mass, the sequence, we decided to start from a completely new approach. First of all, I'm going to refer to genes. Hereafter, when I say genes, I refer to the protein coding genes for simplicity. So, gene length distribution follow log normal distribution. I was working as a postdoc in Canada, and suddenly one day I was exploring pseudogenes, and I found just exploring that the gene length distribution in human follow a log normal distribution. And then I was amazed about this too, because the bibliography had almost nothing in relation to that. There was a paper from 1999 that's showing that protein length distribution, that as you know is the right skew distribution, should follow gamma, perhaps a log normal, but the bibliography somehow forgot this distribution somehow. For me, it was so. Yes, here you have an example with Daniel radio, a model organism, the zebrafish. In blue, the observation, okay, for Daniel radio, that is the gene distribution, and in red, the theoretical one. Only with two simple variables, because with the mean and the standard deviation, the log normal, you build the theoretical one. So first point, we downloaded more than 33,000 genomes, right? More than 33,000 gene annotations for different species. And the first thing is to prove that this is right. This is a log normal, and then we work it with the fits, the log likelihood, we put the distribution in competition. The paper of 99 was saying it's a gamma, perhaps a log normal. So then we try with all the right skew distribution, the Weibull, Campbell, many distributions. And the log normal is the distribution that follows the gene length distribution across the whole tree of life, in the three domains of life. So it's a log normal distribution. Second observation, when we compare the distribution of different groups of organisms, we realize that they increase in size. So in the distribution, you have the log of the length, the mean, and the variance increase with different species, with different groups of organisms. As you see, here is something like the organismic complexity, right? If you compare bacteria, that will be a mammal, no? So obviously the mean increase and the variance increase. So we started to think, and this had to start with Luca. As you know, Luca is the last universal common ancestor, so the grandmother of all the cells, of the species. So it had to have a gene length distribution. And this log normal distribution has mathematical properties. We come back to the central limit theorem. I'm not going to enter because you know about that, but as a consequence of the central limit theorem, okay, the product of random variables converge to a log normal distribution, regardless of the distribution that these random variables follow. So, of course, with enough numbers of tries. So we are observing that we start from Luca, and there are different gene length distributions. And this gene length distribution should be the result of evolutionary operators, I think from the very first species. Okay, so why don't we think, if we know that multiplicative processes end up in log normal distribution, and we also know that the log normals are the typical outcome of multiplicative processes, why not to think that all the evolutionary operators could be modeled in a distribution of multiplicative factors? And we developed a model, a toy model of gene growth based in stochastic multiplicative processes. So we started with the length of, with the genes of Luca, whatever they were, whatever the distribution they had. And on the other hand, we have a distribution of multiplicative factors. Again, we don't care about which kind of distribution we have, because of as a consequence of the central limit theorem, it will converge to a log normal distribution. In which way? The ways that I'm going to show you. In our model, we start with the genes of Luca, and we select one of them. We take the length, and we take a multiplicative factor on the distribution that models the evolutionary operators. The new length will be simply the product of the gene length of the gene and the multiplicative factor. So simple. The rest of the genes that do not change. And starting working analytically with these ideas, we found it first that the mean gene length distributions, the mean of the length of the distributions grow exponentially with evolutionary time. Okay, here we have something that will be like constant because it's the mean gene length of Luca, and this will be the mean of zeta, the multiplicative factor distribution. Second, the mean of the logarithm of the length grows linearly with evolutionary time. So we have like two gene loads. And remember, we have 33,000 species. So we did the fit, and it was going well and doing well. And the issue that we can start approximating these variables, no? These parameters. So this will be the mean gene length of Luca, the mean of the multiplicative factor. This will be the mean of the logarithm of length, and this will be the mean of the logarithm of zeta. So super simple. So basically, if we substitute this time here, we found a relation between the two variables, and this relation is linear between the log of the mean of the length and the mean of the logarithm of the length. That should not happen in principle. So we substitute this time here, and we obtain this equation. So these will be constants, right? And these are a linear relation between the two parameters, between the two variables. Remember, we have 33,000 species. So we took the distribution, the gene length distribution for each species, and we get these values. Let's plot it. And that's what we found. So this linear regression with a goodness of fit of 0.97 is in agreement with the model. We also, after that was for us astonishing, we took the gene length distribution from an early Luca or any early bacteria, and we make it grow, ending up in something that resembles humans also. So at the moment, we have the log normals and the toy model with stochastic, that is basic stochastic multiplicative processes modeling the evolution of the gene length distribution in a very simple way. Next step, big data mining. Simply, if the distributions are so important, let's plot the variance in regards to the mean gene length, mean gene length. And this is what we observed, and this was astonishing for us. So the variance depend on the mean gene length. Okay. So this will be like a scaling law, because scaling law from the microscopic to the microscopic, we have 2 orders of magnitude here, 5 orders of magnitude there, more than 5 or more than 2. So this is a log-log representation. The regression line is also very good, 0.92. But we were not happy with this, because this is not in relation to what I was telling you before. It has nothing to do with the log normal, has nothing to do with the stochastic multiplicative processes. So we started working with the equation. So the property of the log normal distribution and the two Jim Crow laws that we found before. The property of the log normal is something very simple. With the mean and the variance of the distributions of the logarithms of length, you can calculate the mean and the variance of the distributions of the length. Super simple. And these two laws, we work analytically, finding that the second row momentum of the distribution is the power function of the mean Gini length. Right? If this is like that, that will be cool, because the variance, just subtracting here, passing this subtracting, the variance obviously depends on the mean Gini length. But this is only analytically. Second, there is a synthetic with for large mean Gini length. Okay, the second row momentum converge to the variance. Okay, so this will be for largest mean Gini length, so that is vertebrate. And of course, we can calculate analytically the values of the coefficient and the exponent using our equation. So importantly, this slope will depend only in the distribution multiplicative factors, and this coefficient will be depend on this beta, right? And also in the Gini length distribution of Luca. Okay, this is only analytic. What happens with the biological observation, with molecular biology data? We come back to the 33,000 species, and this is what we found. So the goodness of fit now is 0.98. We have the second row momentum against the mean Gini length. The goodness of fit is 0.98, and this will be amazing what I am saying, because the variance will depend obviously on the mean Gini length. The mean Gini length also is a very strong, very robust value, and will be even robust for incomplete annotations. Every point here is a different species, and we have more than 33,000 species, right? I must say that everything that I am presenting now is original. That was never tried before. Okay. Moreover, for vertebrata, the second row momentum converts as predicted to the variance. So same coefficient, same slope practically. Goodness of fit in vertebrata for 0.95. Okay, something else very important. This is, as I said, is a scaling law, and this you probably don't know that many scaling laws in biology for the three kingdoms of life. So there is that I remember, for example, the Cleva law, and there is something but little things showing this kind of laws for the whole tree of life, right? Next, as I am saying, is plotting the different groups of organisms with different colors. We are observing that with the mean Gini length, the groups of organisms, there are certain overlapping, right? But the groups of organisms grows with the mean Gini length, like the observation I was showing you at the very first time of the at the beginning of the presentation. When we calculate it, we come back to the taxonomy, NCBI. This is like the Linnaeus taxonomy from the 1735. And we calculated for every group of organisms the average of all the mean Gini length of the different organisms. And when we plot, it's just the average of the mean Gini length, and they have the perfect order. Archaea, bacteria, fungi, plants, insects, species, aves, non-primate mammalia, and primates. Everything in order, and even protists are here. But protists, I don't want to talk that much about that, because protists is something when people don't know, is the coolest side of evolution. When somebody doesn't know what is a species, then can end up in protists. So you have in protists what you have before as fungi, algae. So protists is a mix of many. So important, the Gini length will be then a very good proxy of organismal complexity. Be careful, what is organismal complexity? I mean, our intuitive view of organismic complexity. Also, we did some more measurements, so we have more even quantitative data. But I'm going to skip that. Next step, we go for the proteins. Well, at the moment, just wrap up. We have the log normal, the model of gene growth as an stochastic multiplicative factor, and then we have the generalized Taylor law, where the variance depends on the mean gene length. So the mean gene length is relevant. Protein, then we go for proteins. Protein length distribution are also log normal. Okay, very well. So this is for zebrafish again. This is an amino acid observation and the theoretical distribution. Again in log normal. The same procedure as before. Same kind of analysis. Log likelihood, fittings, competition of all the different distributions is a log normal across the three domains. Offline, only be careful, only with simple variance. We are not saying that we are in this with this model and everything that we are reproducing evolution. Okay, what we are trying to explore is who was the main motor of evolution. Right? When we plot the distribution, now we have gene length distribution and protein length distribution. Obviously, in prokarya, you have almost a perfect overlapping. Why? Because the protein is mapped in the protein coding gene, the gene, almost perfectly. So the overlapping is perfect. But what happens after the origin of eukaryogenesis? What happens is that the proteins decouple from the gene growth process. So the genes start incorporated, as you know, non-coding sequences, introns. And the distributions that are log normal, they don't overlap. This is the one of proteins, this is the one of nucleotides. Now everything is in nucleotides. Before I represented in this, that was in amino acids, right? That only scaling. One amino acid, you know, one codon, 3 nucleotides. Okay. Then we started working analytically again in the generalized Taylor law, and we find that it should follow the same law. And when we plot, what we observed is that yes, it follows at this in the eukaryogenesis, to the origin of eukarya, and then it's a log normal. But as we know, this was for genes, the second row momentum, and this is for proteins. Now, the mean protein length is not a good proxy for organismal complexity. You see that there's an overlapping, and we can learn a lot about biology from this data, because who has the mean protein length, the largest one? Protists. That is very much unexpected, because we will say, no, mammals, we have the bigger proteins. That's what most people was expecting after sequencing the human genome, right? So as I said, the protein length is not a good proxy of organismal complexity. This was what I did before for genes, now for proteins. And there is no order here. Well, of course, till the origin of eukarya is the same order, perfect. And here there is certain order, but you see fungi has the mean protein length higher than aves or than insects. So, and even you see that the non-primata mammalia is after primates. So no, the protein length is not a good proxy for organismal complexity. Then let's compare the distributions. I said that the mean gene length and the protein length are very good variables for the distribution. I'm going to compare the protein length distribution with the gene length distribution. How? We plotted the mean protein length against the mean, the mean protein length with the mean gene length. And now we don't have 10,000 proteins. We have only 6,500. Why? Because we had data from different databases, right? And what we did is the overlapping between these species, right? Because in proteomes, we have 10,000 gene annotation, 33,000, but the overlapping in a species between them is 6,500 species. When we plot, we found something that looks quite good and quite amazing, that something is happening at 1,500 nucleotides. We were observing that from the data. And yes, at the beginning, you have the perfect overlapping between the proteins and the gene length distribution, the protein length distribution and the gene length distribution in a relation 1 to 3. But the genes continue growing, but the mean protein length stabilize at about 500 amino acids. And this is cool because it's the first time that you can see how the proteins evolve in comparison to the genes. You have 36,500 species. So this stabilizes at about 500 amino acids. That's great. But the issue is that the proteins somehow decouple from the process of gene growth. It does not mean that the proteins do not continue evolving. The proteins were probably tuning. So we have low complexity regions. We have large proteins. So the proteins also in eukarya also develop, but in a different way. But importantly, the mean protein length is around 500 amino acids. Good. So there is an umbral phenomenon here separating genes that almost code for everything for proteins and genes that incorporate non-coding sequences, introns. So this is kind of an umbral phenomenon at 1,500 nucleotides. But is this for real? Because we put it like that. This is like the theoretical. You know that the gene annotation and the proteins annotation in many species are based in the genomes. Genome annotations automatically. So then we went and we got the genomes for the species. And we only got the species that has the best annotated genome, something like 1,000 genomes, the best annotated genome. And now is when plotting the best one is clear that the umbral is at 1,500 nucleotides. So it's an umbral phenomenon, but could this be critical phenomenon? Something else, phase transition like the ones we studied in physics before. For instance, the transition from ferromagnetic material to paramagnetic material, depending on the temperature. The temperature will be the control parameter. When you reach the two-way temperature, you lose the magnetism. You have the easy model with that such a beautiful phase diagram. Here, we have genes that almost code for proteins. Right, so it will be the phase only for genes coding for proteins, and in the other phase, the gene will incorporate non-coding sequences. The control parameter won't be the temperature. In this case, obviously, will be the mean gene length. That something is happening at 1,500. And the other parameter, not the magnetic field, should be the comparison between the protein and the gene length distribution. And we are going to study the amount of non-coding sequences without the gene that can be easily mathematically characterized by this rho. We take for every species the distributions and we calculate very easily the fraction non-coding sequences. So now we need to plot, and this is what we found. And we're super happy because this diagram reminds very much to the phase transition observed in the easy model. Yeah. So then, if this could be a second order phase transition. If this is a second order phase transition, the properties observed in other models somehow could help us to try to understand biology. First of all, if it's a second order phase transition, this happened abruptly. So at the critical length of 1,500 nucleotides, suddenly, abruptly, okay, it changed the phase from a CDA phase to a non-coding phase, right? Okay, again, we have every point is a different species, and we have these two different phases. Moreover, more properties of the second order phase transition. So, you know the critical slowing down phenomena. So perturbation in points close to the critical point will take long time to recover. So that means perturbation at this point, when they are very close to the critical point, will take long time to recover. So they will end up in metastable state. And we will expect to find a cloud of points close to the critical point. And that is what we are finding. It's amazing, that is cool, but that could be because there are many fungi. So let's plot the distribution with the peak exactly at 1,500 nucleotides. But this could be because of many other reasons. So then let's start studying the state. So what we did, we built the fraction, the state. So that means if there is certain row and certain mean length is using a row. So we created a lattice of 100 by 100, and every point will have an state, 0 or 1, like a big chessboard, okay? And it will be black or white. In principle, everything is white, so if there is no species in the point of the lattice, it means that this state is not good. So it will be 0. The state will be 1 if at least there is one species, this point. So 3 variables, row, the mean length, and the state. When we plot the fraction of the state, so it happens that there is a peak exactly at 1,500 nucleotides. And this could be, again, because there are many fungi or whatever. No. We did bootstrapping with all the different groups of organisms. So there is nothing. So the point is that from the properties of and there are many things, but also there is not that much time, and also we couldn't because it's difficult. At the end, everything that we are finding theoretically, we are validating. But it started, it's 15 minutes more. 15 minutes more. Okay, so where I was asking, if this was a second order phase transition, which type of phase transition? Can we say more about this? And this is something we would like to discuss about that. And we propose that this was an algorithmic phase transition. So we are doing a parallelism between evolution and combinatorial optimization algorithms. Be careful. Evolution is not trying to find solutions for nothing. Evolution is searching in a search space, but is producing at the end. At the end, it's adapting. You have new species with larger proteins, but it's not because evolution is trying to find solutions. It's search, then it adapts in comparison to the combinatorial optimization algorithm, right? But designing a protein is known to be an empty problem from the point of view of computer science. And there are search algorithms that has a second order phase transition, but it's called an algorithmic phase transition. And the algorithmic phase transition will be found in the performance of the algorithm. And they exhibit what is called the easy, hard, easy pattern. This is nothing else like a trade-off between the search space and the numbers of available solutions. Okay. I'm going to put an example to try to understand that. Let's play Sudoku. You know, you receive a new Sudoku from the newspaper. You have some numbers, and you have to fill the squares that are empty. Here I represent the easy, hard, easy transition. This will be computational complexity, computational cost. How difficult is to solve this Sudoku in comparison to the search space. And this is the search space goes with the constraints of the system. When the constraints of the systems are very low, that means that you receive a Sudoku that is almost filled in. So you have only few possibilities. Here you have 2. So how many possibilities will be? 100. The search space is very low. To solve this Sudoku is trivial, even if it does not have a solution. On the other side of the board, of the graph, we have a system with low constraints. You mean you receive an empty Sudoku. To solve this Sudoku is again very easy. But now the search space is brutal. How is this possible? I'm going to explain it. If we start receiving from the newspaper Sudokus with more white space to be filled in, more blanks to be filled in, the computational complexity increases because the search space increases. It's more difficult. You have 3, 4, 5, 6. That will be 10 at the power of the number of squares to be filled in, up to a critical fraction. Here, at this critical point, you will receive the most difficult Sudoku to be solved. What happens that if we still remove some squares, and they have to be filling more squares, the search space increase. So it should be more complicated, but no, because the second transition happens, the trade-off between the search space and the numbers of available solutions. Here, the numbers of available solutions suddenly start increasing drastically. And this is what we think that is happening with evolution. This is like a graph. We have an easy, hard, easy transition, algorithmic transition phase that is simply a graph. This will be the second order phase transition that I showed you before. This will be the computational complexity in regards to the mean gene length. So at the beginning, you have LUCA, you have organisms with small proteins. What does evolution? Okay, so it searches, it mutates, and it carry out new species with another protons larger. And it's very easy to do that because the search space is very long. How do you increase functionality? How do you adapt? Increasing functionality. If you have short proteins, at some point you have to increase the length of the proteins to have more functionality. Increase the complexity. I think that is very, very logical. But the longer the protein, the more complex for evolution to design, also to adapt to proteins that are larger, up to critical point at 1,500 newtons. So the longer the proteins, the mean protein length that is characterizing the protein, okay, there is a mapping to the gene. You can think this for the power of the length of the gene, up to critical point. So here, the computational complexity for evolution should be the highest. And we think that very much evolution faces something like a computational complexity crisis, and solve it very elegantly with this second order phase transition. How? With endosymbiosis, incorporating non-coding sequences in the gene. The numbers of solutions then they grow brutally, because then you have the introns, you start combining the different exons, generating much more proteins. Okay? So this will be an elegant solution by evolution, but it's only a draft. Then we define the algorithmic complexity like that, like this tau, depending on the evolutionary time. So we define the computational complexity as the evolutionary time per nucleotide in order to find in evolution for the very first time the gene length distribution with the mean gene length. So in the equation will be so simple. Remember our law that we found for the stochastic multiplicative processes. Then we substitute the time, and we have a computational complexity depending on the mean gene length. That will be this equation. And this equation is nothing else than the graph that I showed you before. And the maximum here will correspond with the critical mean gene length that we were saying that is 1,500. When we calculate the maximum of this function here, is with the derivative, very simple, again, 1,506 nucleotides, so very close to the one we found before. What happens with evolutionary time is pretty much unknown, but now we have the critical time, the critical length. So we can calculate the critical time, and will be at 2.6 billion years ago. Okay. Two main things to think about. We are saying that evolution faces a computational complexity crisis or something like that. And we are saying because the mean protein length that maps the mean gene length increase, and something is increasing the computational complexity. But it could happen that evolution is not exploring globally in the sequence, but locally. So, but we are finding that we are thinking that this is the length of the mean protein length. So it could happen that we are facing, this referred this morning, in one conversation, a catastrophe error, for example, in the transcription or the translation. Even more, I don't think that it's like that. I think that what is happening is that with longer mean protein length, when you have a longer protein, the folding time, and this is known, is longer. You cannot have bacteria with larger, with very large proteins that takes half a day or 1 day to fold, to perform the folding, the origami, the three-dimensional structure, because you need that the whole colony reproduce in half a day or 1 day. So everything should fit. And I would say more. I don't think that is the whole protein length. Probably is simply the length of the domain. But as you know, is the subunit of the protein that folds independently. The longer the domain, the more time it takes to fold. But this is only a discussion that needs to be explored. Okay. Another point, something that I want to comment, and is that we were saying that people is trying to answer the question how, and I think that we are providing a lot of information, observation, biological observation of how this is happening. But if we think now, we are seeing that there is an stochastic multiplicative processes, and we are reproducing how the gene net distribution evolves. This is very interesting. Why? Because there is no natural selection there. So this can blow the mind to many people. In regards to the gene net distribution, there should be universal law or the physics or whatever that make these distributions to grow like that. But this is what we are reproducing. I'm not saying that we are reproducing what evolution is doing from a point A to a point B. What I am saying is that we are modeling and we are going from a point A reproducing the same thing that evolution did. So the natural selection visible should not be needed, but this is very controversial because there are many streams of thinking, but this is what we are observing. With that, the conclusions. So, first of all, gene and protein length follow log normal distributions across the whole tree of life. Second, the gene growth process could be explained as a result, not as a result. We come with a toy model, not that we are reproducing what evolution is doing, but the main motor of evolution will be an stochastic multiplicative processes. And the very same one for the protein, eukaryogenesis. Third, we found a scaling law, a generalized Taylor law, okay, for the whole tree of life. The variance depends on the mean gene length. The mean gene length is a very strong variable. The proteins grow with the genes up to a threshold of 1,500. We saw how the distribution grow in comparison to comparing the mean gene length, protein and gene. We found that we think that is a second order phase transition separating the coding phase from the non-coding phase in regards to the content of the genes. And we propose that this phase, that phase transition was algorithmic. Phase it by evolution when adapting to proteins with a larger mean protein length. And with that, I would like to acknowledge, obviously, my co-author. This is not my work. This is the work of 4 friends, of 4 colleagues that we worked very hard doing slow cooking science for many years. It's nothing that arises like from one day to the other. And also, these are my people and collaborators that I had around, and I was discussing about that and more subjects, the references of the papers. And with that, thank you very much for your attention.
Thank you for the talk. Mario, there.
Thank you. Hi, can you hear me? Okay. So I have a question. When you have computed this intrinsic time, tau, you have computed this intrinsic time called tau, which is t divided by the mean length.
Yeah.
And then you substituted time according to the average of the length scale and so on. I have the feeling that you're using the same laws that you have used to plot the original data, the proteins versus the gene length, to compute this intrinsic complexity time and so on. Is there another way to measure complexity which uses different information or different data set or a different metric?
So we define the computational time like that. I don't see the relation of what you are saying. That you have the impression that this is like that. Evolutionary time. Okay, let's start by evolutionary time. It's not my cup of tea. First of all, evolutionary time, just think in the most recent event. So what do we know about history? what we have in the book, no? And in the books of history, if we go for the recent history, last 200 years ago, we know a lot about history because we have the books. But if we go further to the Roman time, before to the Etruscs, so it's getting more and more complex because we have less documentation. Doing a parallelism now in molecular biology, the book, the evolutionary time, thinking the most recent event for humans. Well, we have our cousins, Neanderthal, Denisovas, and so on, but the speciation with chimpanzees. When I was starting to teach at the university in Mainz 10 years ago, the average of the publication was in 6.7 million years ago, the speciation. Now you go and check, and it's 6.4. So there is 0.3 that every decimal is 100,000 years ago. And this is for the most recent. Of course, the history of life on Earth, the history of the universe, then go to the other side, the origin of the universe. And then you have the Hubble tension. Okay, this very much precise, but still there is a tension. The Earth, yes, because the evidence is very strong. The origin of life, 3.7. The origin of Eukarya, very much unknown. So, and then you go and you say, fungi, 1 billion years ago. Why not 1.5? Why not 1.8? So, evolutionary time will be taken with a grain of salt. So, I try to avoid, but what we saw is something analytically, is something else. It's extracted from the equation. So, if you have the feeling, I don't know why, but in principle, I don't see it. So,
Christian?
Thank you. So probably by my ignorance, I am starting to miss the relation with the emergence of eukaryotes. I mean, eukaryotes have a certain complexity, but I can imagine there are various types of complexity. You discussed a particular type of complexity, but why is that related to the emergence of the nucleus, for example?
Well, that's what you have. Okay, so the idea is that at the very beginning you have a simple world. So the cells were simply unicellular. We don't have organisms like us. In our bodies, we are composed but many cells that are interacting between them. I think we'll say that this is much more complex. It's very difficult to talk about complexity. What is organismic complexity? Honestly, I cannot say. I have an intuitive idea and some quantitative also data. For example, the numbers of tissues. No? The numbers of tissues an organism will be an indicator of the organismic complexity. But this something, as I said, is something very subjective. What is organismic complexity? When the Human Genome Project was carried out 25 years ago, we were expecting to have 100,000 protein coding genes. Protein, why? Because we cannot have 20,000 like elephants, worm, because we are much more complex. But we had, we had at the end the very same numbers of proteins. So how to define complexity? I totally agree with you that this is so it should be taken with a grain of salt. But what is clear is what we are observing with the gene length distribution, that they grow up with time. The data is showing that definitely. But this is you're totally right. So it's that should be taken with obviously with the grain of salt.
One more last question.
Thank you. Very nice talk. So I have many questions, and my mind starts to all about all this.
I know you keep that feeling.
I don't know if I correctly understand your research, but for me it's very strange that this phase transition occurs in the sense that, if I correctly understand, you have some multiplicative law for building new proteins in new system. Maybe, I don't know if virus has some role in this type of growing in this species, because virus they put their part of the genetic code into the genetic code of the cells. No? It is well known. But for me, it's very strange that sometimes, or some length, of the proteins start the transition, and if I correctly understand, no larger proteins can be built with this multiplicative mechanism.
In average, so mean protein level.
On average, okay?
So larger
And
protein.
you said that this is due because for this length, the proteins to fall and to be functionally active is very hard, no? If I correctly understand. So then my question is the following. Do you think that, for example, with this type of artificial intelligence tools to build proteins, other type of proteins, no? Because I think the phase space of proteins will be larger than this size. So maybe there are some physical constraint that we can not build functional protein using, for example, these artificial tools.
Okay, not for the protein. So the mean protein length is stabilized at about 500 amino acids, right? But the proteins continue evolving. So they decouple from the gene growth process, right? And they start refining. For example, incorporated low complex regions. What do these low complex regions, for example? Think in the quaternary structure, the protein complexes. That's a clever way to build more biomolecular machines. Even if you have introns, you have RNA, and this RNA also can couple to the protein complexes. So the proteins continue evolving in another way, but the proteome, you take the proteome, and the mean protein length is exactly the same. Because of the properties of the log normal, you have a symmetry. Long next to short, and that's symmetry. If you build a long protein, you will have a short one. If you have a long protein like human, we have 15, that is 30,000 amino acids. It takes, I don't know how many, half a day to do the folding. Or we have neurulin, 7,000 amino acids. So we can carry out longer proteins, but we cannot have the mean protein length made by longer proteins because we think that it's because of the folding time. And at the end, the solution of evolution was amazing because the solution is like encoding. At the beginning, what you have is a linear programming, okay? But evolution invented the if condition. If this happens, no, I select something else. Super smart. So I think that they cover everything. Regarding the artificial intelligence, I think that there are what I understand, what I am seeing, I think that there are fundamental laws of the physics, yes, that basic in chemistry, basic in whatever. But something is happening that is making this, growing this gene length distribution with mean gene length at the very same time in the whole evolution. There should be fundamental laws there, like, I don't know, like gravity, like things that are there, and we don't understand.
Thank you very much.
Thank you.
So we have our next speaker, Mario Castro.
Can you hear me?
Madrid.
Is this working?
Yes.
Yes. Okay. So, thank you for being here. This is unlike Miguel Angel. This is my first time at the Granada Seminar, so I'm really happy to be here in this legendary meeting that I haven't been able to attend, typically because I'm always teaching on September. But probably I will return. So I come this morning with a student's mindset because this is my first time. So I've been taking notes like crazy. So I've been enjoying a lot the previous speakers' talks. Okay, so these are my collaborators. Today I'm going to talk more about philosophy. I'm going to try to model how we do science rather than the specific models. I'm going to land on specific systems, but I think I want to open somehow the discussion of how physics works and how modeling works. Why? Because these days we are somehow under the impression that everything comes from data, and sometimes physics is getting old-fashioned, but I think we still have something to say and why things that we have used in the past are still working in the future, are going to be working in the future. So I borrowed this first part of the title of the Granada Seminar, this part of new tools and methods. I'm going to talk about three tools: the old, the newish, and the brand new. The best is the old, and then the newish, and the worst one is the newer. But basically what I want to do is start with some philosophy, then land on some specific case studies, and then finish with some reflection about how Bayesian statistics can guide typically the way in which we do science and which decide which theories are better than the others. Okay, so when we talk about simplicity and complexity, we know a lot of tools. I did my PhD thesis in the 90s, so okay. So I did my PhD thesis in the 90s, and pattern formation was an issue there. The normalization group was everywhere, so I learned from one of the people in the audience. I know your names since I was a student, and I'm in love with all these tools that you all know. But somehow, maybe because we are being under the threat of this big data and deep learning stuff, I'm starting to thinking, okay, these tools are great, but where is the data? So when we talk about these tools, typically we do some fancy calculations that take us pages and pages, typically a normalization group. But what is the data here in this analysis? So we have very good theories that explain why simplicity and universality emerges, but we are not taking, my feeling is that we are not taking data seriously into account. So I'm going to start with a very hard problem. I'm going to ask your child brain, what do you see here? Probably you see a tree in a blue sky, well, not so blue, but in a blue sky, and then you see something behind the tree. Okay? This could be a rectangle, a box, or whatever. Okay? Probably you all are seeing this right now, right? But what if I ask you, okay, how many boxes are behind the tree? How many gray boxes? Then you start thinking, okay, maybe there are more than 2 boxes. But it would be a matter of coincidence if you have just 2 boxes, but it turns out that the border of these boxes are hidden by the stem of the tree. So somehow our brain can distinguish between a simple model and a model that could be, but it doesn't sound very possible. Okay, so I'm going to put some numbers in this sort of calculation because I think this is the way in which physics typically has decided which models are better. Okay, to do that, I have to introduce briefly what is the Bayes rule. Probably you have been exposed to Bayes rule in statistics, but statistics provides a very narrow view of the Bayes rule, of the Bayesianism in general. Why? Because typically we talk about events. We say the probability of event A given event B, but the Bayes rule, somehow in my view at least, I'm a Bayesian, and my view is that it measures our degree of belief in something. So you can plug whatever you want. So you can plug events, but you can plug anything you want in this equation. So for instance, we can plug a theory, a hypothesis case, you can plug data, and you can plug the parameters of the theory. If you have this open mind and you think that everything can be plugged into the equation, then we can reread this Bayes rule, because we can say that this prior probability is nothing more and nothing less than what we know about the parameters or about the theory before seeing any data. For instance, imagine that a new pandemic comes in, I hope not, but a new pandemic comes in a few years, and then we want to estimate the infectious period. And we say, okay, I don't know, but probably more than 1 day and less than 40 days, because this is quarantine and this is typically, nobody has seen an epidemic or an infectious period as long as 40 days. So this is our prior belief before seeing any data. Then we have our model. This is our theory, the likelihood. This is where we plug the parameters. If we knew the parameters, what are the predictions of our theory? And then if we multiply and normalize, we get the posterior distribution that basically tells us what is the probability of those parameters according to this theory and according to the data, the experiment that we have carried out. Okay, so this is Bayes rule, but applied to theories and parameters. Okay, it's important to say that despite normalization, it's important to say that we can plug, as I was mentioning before, anything here. So you can bracket parameters and theories. So you can marginalize for different theories. And actually, you can use Bayes rule to compare theories. So instead of saying this theory is better than this theory because it's more elegant, okay, this is what string theories say, or this theory is better than this theory because it's more beautiful, we can say this theory is better than this theory because it's more plausible. So simplicity is a matter of plausibility. And plausibility is probability. Okay. So let's go back to our physics problem. Theory number 1. Of course, we have an infinite number of theories, but okay. Theory number 1, there is just 1 box behind the tree. This is what your brain told you probably. Theory number 2, there are 2 boxes, and just by a matter of chance, we have the 2 boxes hidden by the stem of the tree. Okay. Let's compute some numbers. So let's define this quantity L, which is L1 plus L2, which is typically the length scale left by the tree. So everything that we can see. So model 1, we need to, this is a rectangle, so we have to say we have to allocate 3 corners at random. So in the space of all potential boxes that we can allocate, we have to put these corners. We have this scale L, which is free to us. So basically we use a random distribution, a uniform distribution, then we can say the probability of this data according to this model is 1 divided by L cubed. If we have two boxes, then we have to allocate 6 corners. And of course, the probability now goes like 1 divided by L to the 6. So if we divide both probabilities, we have this scaling argument that tells us that the larger is L, or the other way around, the narrower is the stem of the tree, the more likely is the probability for a model with just one box. Okay, that's what our intuition tells. Okay, you may be thinking, okay, where is the physics here? Okay, this same argument was proposed by Harold Jeffreys in this magnificent book that everybody should read called Scientific Inference. And basically, he asked the same question with this Galilean experiment. So this is the original experimental device by Galileo. And imagine that you have this inclined plane, you have different observations from T1 to Tn, and you have two theories. Theory number one, the usual one, the one that we teach our first year in physics, and model number two, which is undistinguishable. Why? Because if you plug here the observation times, T1 or whatever, this term is cancelled. So basically you have both models, a couple of theories that are not distinguishable to each other. But this is model with one box, and this is a model with n boxes. And now we have an argument why we prefer this theory than this theory. Why? Because the probability, the likelihood, or the posterior distribution for this model is really small. Okay, this is not very likely because any deviation from this observation times is going to throw away this model. Okay, so keep this idea in mind. I will return to this view of information, belief, plausibility, and the space of possibilities later. Now I want to discuss a couple of different approaches to discuss why simple theories, why what we call toy models or spherical cows, are more, let's say, preferable in most cases. So keep this message in mind, okay? This is not doing good models in physics or simple models. It's not about laziness. We don't use toy models because we are lazy. We use toy models because we know how to interrogate them. And now we have another argument. Toy models are good because they are plausible according to the data. Okay, let me discuss these case studies. I'm going to start with something that should appear. It looks like a publication, but no, this is a preprint. It should appear this month, but it's taking longer than I would like. And basically what we have done here is try to dismantle this whole enterprise of using the Lotka-Volterra and similar models to understand the microscopic dynamics of the microbiome. So this is the narrative in the field, basically. So you have now RNA sequencing, so you can analyze feces, for instance, in the gut, different species, different ecological environments. You can do that at different times, and you do that at different times, then you have this time course. Okay. Now you can plug these time courses into a model. It can be Lotka-Volterra, or it can be any model with the resources or whatever. It can be as complicated as you want, but the argument is more or less the same. The good thing is that now you do some fitting, and when you do the fitting, basically you are extracting these parameters beta, which are called the interaction matrix, or the interaction network. Okay, this interaction network is really nice because it's telling you that, for instance, the orange bacteria are related in a positive way with the yellow one, and the blue bacteria are related in a negative way with the yellow one. And this drives to what they call personalized medicine, in the sense that, okay, if you have this problem and this problem is caused by the yellow bacteria, I now have the instruction manual, so I can take these betas that I have estimated. I know that I have to feed, or I have to start with the orange ones, and I have to feed the blue ones. Okay, I'm going to say later that this cannot be done. Okay, why? Because if you take a look at simulations of the Lotka-Volterra model, these are a couple of examples with 4 and 5 populations, but you can imagine that this is almost the same. You have typically two kinds of pattern: oscillating stuff, so things that, like in the original Lotka-Volterra works, something that is oscillating, a couple of species or whatever, or you have something like this, in which you have a growth, then you have a peak, and then some sort of stabilization. Okay, I don't know you, but I've been working in many different fields since my PhD. I've worked in epidemics, ecology, T-cell receptor dynamics, condensed matter physics, and I've seen this picture many, many times. And again, going back to your child brain, in the same fashion as you have seen just one box behind the tree, probably you've seen this curve many times in your life, okay? Electronics, wherever. So when we statistical physicists see something in many fields, we start to thinking about universality. So maybe there is something universal here. We want to understand why. So let me follow in this very simple calculation. So if you see this curve, maybe you can count not how many points do you have in the curve. In this case, you have an infinite number of points because it's a smooth curve. But in an experiment, you can have as many points as you want. But the question is not how many points, but how many pieces of information. So here you can count. For instance, you can estimate the time to the peak, or that could be related to this exponential growth. Here, for instance, Then you have the steady state, the distance from the steady state to the peak, also the duration of the peak and the initial condition. So basically, whenever you see in the future this curve, think about me and see, okay, you have 5 pieces of information. If your model have more than 5 parameters, then maybe you're overfitting. Okay, so a few months ago, I was playing around with some other equation. I end up with this crazy function that fits pretty well this sort of estimation. Here and here you have just 5 parameters. If you use this equation and you randomly select different parameters, you can have almost anything you have seen in ecology or epidemics in the last 20 years. So basically, with 3 parameters, you can have these orange ones, which are sigmoids. With 4 parameters, you can have these magenta curves, looks like epidemics. Okay? These are asymmetric. And with 5 parameters, you can have these bumpy things that I've been discussing. So basically, you have a universal law, let's say, for this sort of model. As for Neumann says many years ago, this means that with 5 parameters, I can fit your curves without any theory. So if you want to propose a theory, you have to be more convincing that a good fit. Okay, let's do some calculations. So if you have n species in your local Terra model, then you have, according to this very simple model, you have an agnostic theory that tells you that with 5 n parameters you can explain the data. No physics, no biology here. Okay, no coupling. So you can explain everything with any type of coupling. If you have local Terra, you have n times n plus 1. So above 4 species, then you cannot tell if those parameters are real or not. Why? Because you have much more parameters than needed to explain your data. Okay? So, okay, this is a kind of back of the envelope calculation. Can this be formalized? And the answer is yes, and this is the newest theory that I was talking about. This is a theory that James Sedna, that probably you all know from statistical physics, is a brilliant guy with many crazy, in the positive sense, crazy ideas. And he's been pushing this idea of the sloppy model theory. So basically the idea, and I don't have time to explain this, but the idea is that if you measure physical information matrix, which basically tells you, remember this likelihood that I mentioned before? Okay, the physical information matrix is more or less like the Hessian of the likelihood, according to the parameters. Okay? It's related in that way. So basically that matrix is symmetrical, because it's a continuous function and you take in second derivatives. And then you can define a metric. And when you have a metric, then you have used all the machinery of differential geometry. And these guys came with this very interesting idea that if you see information, you see the physical information matrix as a defining manifold in the possible world of all the possible parameters of your theory, then you can draw geodesics. And those geodesics are leading you to boundaries. And those boundaries, by definition, have one less dimension than the original one. So if you follow this geodesic, then you reduce one parameter, then you do again and again and again, and then you have a reduced model with less parameters. So this is basically the idea. So we apply this to different simulations. This is another example. This is interesting because this case is chaotic, so this simulation provides a chaotic simulation. So this reduction actually depends on the window span that you are using. But basically, if you do that, you apply this method to this model, you start with 20 parameters and you end up with 7 parameters. The fitting is not that good because the orange one is spoiling everything else. But if you start again and you forget about the orange and you say, okay, I'm going to use the orange as a linear combination of the other 3, then you have 7 plus 3, 10 parameters. So this is enslaved. This is a kind of adiabatic elimination. Then with 10 parameters, you can explain the data, almost indistinguishable from the original data without noise. So here are synthetic simulations. What does this mean? This means that basically you can drop most of the elements of this interaction matrix and everything is almost the same. Maybe you're familiar with this work by Miguel Angel last year that basically they said the same coming from a different viewpoint. So they say, okay, the interaction network matrix is sparse, meaning that you can throw away most of these interactions and everything is going to be the same. But now here we have a couple of possibilities. One possibility is to say, okay, this matrix is sparse because biology is sparse. In the sense that bacteria, maybe they are competing just with one of the species. So they are very selective. They don't compete with anybody. So they have their own enemies or their own friends. Or you can say, okay, this is just an epistemic problem. So this sparsity comes from data fitting, from inference. Okay, and this is much more complicated, but this is more, aligned with what we think about doing a theory. So imagine that those RNA sequences trajectories are the trajectories of molecules in a gas, and I can tell you, okay, write down Newton's equations and now infer all the parameters in those Newton's equations. This is crazy, right? And we discovered in the past that maybe using pressure times volume proportional to temperature is better than using this approach. So we have here an interesting conclusion is that this reduction method is not just a way of criticizing complex theory. It's also guiding us and is telling us that maybe this is not the right way. Because if you cannot tell between a complex theory and a more simple theory, and complexity is a huge word here, so I'm using complexity as the number of parameters that you need to use. If you cannot tell between each other, maybe you have to use a different approach. So maybe we need to define a thermodynamics of the microbiome rather than a Newtonian mechanism microbiome. We are trying to do that in Madrid in a group, but it's not easy because we don't have a pressure and we don't have a temperature. So stay tuned. And you're welcome to come to Madrid to discuss this stuff because this is an emergent problem. Okay, you can apply the same to these typical oscillating things, and the conclusions are the same. So I'm not use this model because I can do that. So you can do that almost any simulation with logical term. Okay. Another example, we took this paper by Saul and Susana, and then we applied the same idea. Here you have the typical epidemic model. Here we have many compartments because now we also have reinfections and we have exposed, and we have two sets of in the population we are divided in two groups, the elder and the younger. Simple. So we have 10 parameters per group, so we have 20 parameters. Okay, we did the same thing. The only thing that is important here, and this is related to the discussion about the tree and the gray box, is what about the information? How many information do I have in my problem? So the thing is that typically in epidemics, we only measure infected and reinfected. So basically you only have these four curves. We don't have all the information of the problem. So if you take a look at this magic formula, then you can say, okay, we have two sigmoids, 3 parameters each, one bumpy thing, 4 parameters each. So with 14 parameters we should do. So we don't need, maybe we don't need 20. So let's figure it out. And actually we found out that we don't need 13. So with just 7 parameters, you cannot tell the difference. So you need a good eye to see the difference between these two fittings. They are almost the same. They are not the same. I will share the slides later if you're interested. And you can see some differences inside the cycle. Okay, this is really good. And I'm using perfect data. So I'm playing God here. I'm not using any noise, experimental data. So I'm playing with synthetic data. Okay, so the conclusion is the same. If your data is poor, if you don't have enough pieces of information in your data, why your model is that complicated? Unless you want to use the model to generate hypotheses. I'm good with that. But my concern is with a division of reality to those parameters. So if you do a fitting and your fitting looks very good and you see that everything is fitting perfectly well, you cannot tell that that parameter measures the infectious period. Why? Because that parameter maybe is irrelevant. You can only measure a smaller number of parameters there. Okay, what is interesting in this case is that if you take a look at the final model, you see that the elder and the younger are uncoupled for some reason. It turns out that the model for the younger is an SIR model. The other one doesn't have any name. It's a kind of integrated ACIR model. But what is interesting here is that this model is emerging because the data is so poor. Because the curves are so simple that almost everything works. Okay, but that may explain this observation that came in this nice paper back during the pandemic that said, okay, the unreasonable effectiveness of the SIR model. Maybe it's not unreasonable. Maybe it's a matter of the poor quality of the data. So if your data is poor, if you only have a bumpy thing, then you cannot expect to have a very sophisticated model. Okay. Another example. I can share the slides later. Another example. This comes from astrochemistry, and here this in collaboration with Marina and Marina's Jacobo's student. Maybe some of you know Jacobo. And here the idea is to study what is going on in interstellar dust. And here the field is much more difficult because you cannot do experiments. Basically everything is observational, and people has been observing the spectrum in the interstellar dust, and they've seen some species appear here and there, and they try to use the reactions that we measure in the lab on Earth to infer what is going on in interstellar dust and to make some guesses about how the gas are condensing or the galaxies are expanding away. We tried to use this method by Sedna to this problem, but it didn't run. Why? Because we are spanning several orders of magnitude in concentration, but also in time, so many millions of years. So the model couldn't run. So we invented a new model that we jokingly named it Fisher. And the idea is the following. So what is going on inside this Fisher information metric is the partial you can write this as the partial derivative of the likelihood according to each parameter. So basically, this is a sensitivity matrix, not the sensitivity of a function, but the sensitivity of a probability, which is the likelihood itself. So we thought, okay, maybe this is like doing sensitivity analysis, but instead of doing sensitivity analysis in the usual way, in which you fix all the parameters and you just move one and see what happens at the end, and then you fix this, move this one, happen at the end, what we are doing here is using the eigenvector of the lowest eigenvalue of the Fisher information matrix to know how to move them all at the same time. Why this eigenvalue? Because this is the one that is telling that that combination of parameters is not informative. So you can tune those parameters and nothing happens. There is no information there. So you can do that, then iterate, do that, then iterate, and then you end up with the same model reduction as in the works by Setna, but you can program this with ChatGPT in a couple of prompts, and it's very easy to understand. Marina did the programming herself, but you can do that with ChatGPT. Okay, so we applied this to one of the models, which is called the phosphorus network, and we started with this network with 14 parameters. We decided to stop with a model with 6 parameters because the error here is the relative error is 10 to the minus 6%. No, sorry, 10 to the minus 4 in percentage, so it's really small. And the interesting thing is that the resulting network, which is this one, we showed this to Izaskun, which is the expert astrochemist here in Jacobo's group. She said, okay, this makes a lot of sense. Why? Because we have not only reduced the number of parameters, we have reduced also the number of species. So some of the species in the original model are not present here. And she said, okay, this makes a lot of sense. Because I've been thinking for this for a long time. She does this ab initio quantum computation. So basically she tries to measure the kinetic rate. And she said, okay, it doesn't make any sense that with those low concentration in interstellar dust, those reactions are there. So again, this is not just a matter of being lazy or having a simpler model. This is a matter of guiding science. Before we have learned that the sparsity is an issue. Okay? The sparsity is relevant to understand microbiome, and that can lead you to think that maybe we have to change the focus. Instead of using differential equations, use ensembles or whatever, probabilities. And this is telling us that maybe, I don't know, we don't need all the species that are related to phosphorus here on Earth. Maybe we have to use something that is relevant out there. Okay? So take-home message number 2, you can use information geometry or this lazy version, this Fisher version, which is based on information geometry, but it's much simpler, to guide you, to put the conversation elsewhere. So instead of thinking, is this model good or bad, maybe the question is this the right approach to this problem? Okay? So let's go back, and I'm finishing now. Let's go back to base rule again. Let's put everything together. Now, let's try to see Lotka-Volterra again, but instead of using this fancy information geometry, let's use base rule. Why? Because it's been there for 4 centuries, so maybe you can use these ideas of degree of belief to understand modeling, to understand how good or bad is a physical theory. So basically, now we can have a definite answer about information and relevance. So if you have some data, some experimental data, and then you have some prior information, which is the blue curve, and then you plug the data into this Bayesian inference machinery, and then you see this posterior, then you say, okay, then this parameter is informed by the data. Good, this parameter is relevant, at least according to this data. Sometimes you learn something, but you still have a lot of uncertainty, and sometimes there is no information. So basically, you don't know anything here. And this is connected with this idea of the sloppy model theory. In this case, this is a sloppy parameter. Why? Because there is no information there. So you can do whatever you want. So you can move these several sigmas with respect to 0. Positive, negative, nobody cares. But this is a stiff parameter in the sense that if you tweak this parameter, everything is going to change. So this parameter is important here. And this is the idea of Occam's ratio. Okay? So simplicity, again, is a matter of plausibility. A simpler model is not as simple because we are lazy or because this is elegant or because nature should be simple. It's simple because experimental data is telling us something. So theories in the vacuum are nonsense. So we have to somehow have a trade-off between what the experiments are telling us and what our theory with this experiment combined is telling us. And the answer is the posterior distribution. Okay? So just to finish, an interesting observation. So what happens if you generate synthetic Lotka-Volterra trajectories, then you increase noise, and then you try to use Bayesian inference to produce what they call the posterior predictive basically is you have the posterior of all the parameters, then sample those parameters and see if this model captures reality. The answer is yes, even for large noise levels. You can see that the theory is doing a very good job. But take a look at the parameters. When noise is small, everything behaves as it should. So the curves are perfect. So basically, these red lines are basically inside the 95% of the posterior distribution. Basically, you are recovering what you plug into the model. When you increase the noise a little bit, something weird starts to happen. Some of the parameters are shifted towards zero, and they are not predicting even the real parameter that we put there. And some of the parameters are changing sign. These magenta bars basically are telling about purple bars are telling us that some connection that you behaved it was a predator prey, maybe it's a prey predator. Okay, because you are changing the sign in all elements of the matrix. And this is crazy. And you can be asking, okay, why the Bayesian inference model, if it's the gold standard, is not capturing my real value? And the answer is because you have introduced noise, and noise means information dissipation. So adding noise means that we dissipate information. So you have a fixed amount of pieces of information that you have to share between all the parameters. If some of the parameters are irrelevant, and I proved before that they are irrelevant, those are going to go to zero. Why? Because you have to put the information where it's used. Okay? If you increase noise even more, this is more crazy. So more parameters are changing sign. More parameters are, this in red, are completely wrong. So basically, adding noise means that any hope that you can extract the microscopic information, this microscopic network from the experiment, is doomed. Why? Because noise is spoiling things. Okay, so even if you are happy because you think that you can predict the model well, this plan is completely out of your hands because noise is spoiling everything. Okay, you can say, okay, but maybe if you cut so many parameters, you are going to spoil things down the road. So maybe if you have new data, maybe the full model is going to do better. This is not the case. So we did an experiment. We start doing inference after we train with up to time 3, and then try to see what happened next. And this is the relative error. You cannot see it very well, but these lines, which are thinner, are the lines produced by the full model, the model with the 20 parameters. And these lines here are the lines with a reduced number of parameters that we have provided with this feature information retrieval. So the relative error is even small, so it generalizes better. And we know that. We have seen that so many times. So you have a large degree polynomial, maybe your fitting is good, but you're going to generalize very poorly. And this goes back to this idea that model 2, according to Jeffries, is very bad. Why? Because it fits perfectly, it fits perfectly the data, but when you shift away from these observations, it's going to generalize poorly. So this is it. First conclusion, whenever you see this sort of curve, count how many pieces of information do you have there, and accommodate your model, the number of parameters of your model, to the real information there. The second thing is that, to me, Bayesian inference is scientific inference. This is not my idea, this is Jeffrey's idea. And the idea is that probability is the language of science. So basically, your degree of belief in a theory, your degree of belief in a parameter, for instance, has to be pondered by the data and the probability that the information that you can extract of that data. If you cannot do Bayesian inference, because sadly this is not plug and play, we are in this era in which doing deep learning seems easy and doing Bayesian inference seems hard. It's crazy, but it's like that. Sometimes you can use some proxies like this Fisher idea that I say is very simple. It's basically computing the spectrum of the physical information matrix iteratively until you reduce some of the parameters. And then probably the most important thing, this is not just an apology of simple models, or this is not a criticism of complex model. This is a way to say that we have to read between the lines. So we have to learn the clues that the data is putting in front of us. So our theory, in addition to the data, has to tell us that maybe we're using the wrong approach, and maybe we have to go back to the classics and do things better. Thank you very much. If you want to know about the details, I have a very short video in my YouTube channel explaining the more mathematical details. And if you want to have a fun time, I just speak Spanish. Next Wednesday, the third season of the Spherical Cow podcast is going to appear, and we're going to talk about Bayesian, rule, and entropy. So stay tuned. Thank you.
Fantastic. Questions?
Yeah, very nice talk. Thank you very much. And so I was thinking about what you said at the end that complicated models do not generalize so well. And yeah, of course, I found this as well in academics and so on. You try to use the best model you can. It doesn't predict the future at all. So, but don't you think that is because when you have more data, we cannot find really a model that really fits this data. And therefore, we have to deal with a simpler one, which is it's wrong, but it captures everything.
Yeah, there are two things. One is that, and the other one that I was prepared to answer is this paradox that they call the second, I think it's called the deep double descent. That this is typically what we see in our traditional models, that if you go too far, you can overfit. But for some reason, in deep learning, something magic happens. So you increase the complexity of the model, the number of neurons, the number of layers. Somehow things start to work better again. And I think the brain is doing almost the same. So I don't understand this. I don't have a Bayesian answer to this. I have some guesses, and it's one of the things that I would like to explore in the near future. Because maybe what the success of deep learning is related to the fact that these learning algorithms are finding models, and we are not able to propose those models, but these algorithms, yes. And connected to the previous talk, I think this has to do with this idea of the Sudoku, that when you have very little constraints, because you have a huge number of neurons, huge number of layers, you are not finding the solution. But you're finding so many paths of solution that you end up solving a problem in ways that your initial simple model couldn't do. So there is a huge, I know, there's a lot of things to explore there, which are far away from the way we do physics, and it's interesting from a physical point of view. But, yeah. Thank you.
So I've been sort of uncomfortable with these ideas ever since reading the original papers by Jim Seth done. Try to. So first of all, it seems to imply that the goal of making physics models is to fit some data curves. So okay, it was applied to some biological systems. You measure 3 variables out of a system that has 100 variables. So not very surprisingly, from my perspective, most of the parameters in your model, which has way too many parameters compared to your data, is going to be irrelevant. So you shouldn't do that. But maybe, you should try to discover if that the I mean, so let me state it in terms of the ball rolling down the hill. We would never think that a reasonable physics model had the specific times of the measurements as part of the model. It violates our way of thinking about how you construct physical models of the universe. So when we make Volterra models, we're not violating that principle. That is a reasonable assumption, in certain contexts, to say that species interact with each other. And if we measure 4 variables, but we have 27 different parameters, then we're not going to learn many of those things. And then the use of that can be to figure out what is another experiment that will tell us about the parameters that we were unable to determine based on the data so far. So I'm just somehow...
Yeah, I agree with you, and this is related to this idea that you have more parameters than pieces of information, so maybe you should do more experiments and extract more data.
You can work, but you can use your methodology in some sense to learn what new experiments would be most informative about things that you think actually might be real physical parameters.
And there's another use of modeling that there is not data fitting, of course, which is... Yeah, no, there is another use of models that is not data fitting. I agree with that. It's generating hypotheses, or what if we confine everybody in a pandemic? That's a useful approach to the model. But remember the other thing that I told at the end, that noise induces information dissipation. So even if you do more experiments, sometimes noise is... You cannot reduce all the noise in the experiment. So maybe you're doomed that some of those parameters cannot be measured. And if you have many parameters that you want to infer, and you need a lot of experiments, maybe, again, this is a signature that you have to change the approach. Maybe you have to use thermodynamics instead of mechanics to understand the microbiome.
Yeah. Thanks for the very nice talk. I had a question similar to this last discussion, because I think during the talk there was some kind of emphasis that models that maybe have less parameters in the micro canonical way are simpler, but sometimes this maybe it's not the case, because if you think, for example, on the Lotka-Volterra model, in which is fully connected, you have n square parameters. But if these ones come from a Gaussian distribution, you only have 2 moments relating them. If you have, for example, a sparse network with all the weights the same, you can have very few links. But the distribution that generates this has way more moments, and it would be way more difficult to.
I totally agree, but
So
you're doing thermodynamics then, because
Yeah,
your Gaussian distribution with the parameters is an ensemble somehow of an ensemble.
Uh, the question I think sometimes is, do you think that we should still try to get some information in the microscopics? Do you think this is useful, or do you think that really maybe some of the lessons that we have to go from this is should really do thermodynamics when we are in this kind of things? How can we decide when to go from the microscopics and when to go for thermodynamics?
I would say that the larger the data set is getting, and the more thermodynamics you should do, because if you think about it, with this RNA sequencing, we have huge data sets, gigabytes, and we think that that's good. But imagine that we have this technology back in the day, and we have a machine that measures all the trajectories of the particles, and we are so happy because now we can do a theory, a Newtonian theory, because we can measure those trajectories because we have this amazing machine that measures all the trajectories. Would you go through the Newtonian way, or do you still go through the statistical mechanics way? We go through the second one.
Just very quickly, maybe it has been said, but is it not important to really be clear, make a clear distinction between what we call theory and what we call model? And these are not the same, right? So if you start by explaining your Bayesian inference for a theory, and then your examples are really for models, that's not the same. And also, there is a difference between the general scientific methodology and problem of data fitting, right? I mean, this is quite a distance. So I think that probably you agree that if you want to speak about what is a good theory or why do we like theories, it's not just a matter of plausibility with respect to data. I mean, there is much more in good theories related to in what sense are they unifying, in what sense are they strongly connected with another theory which is more down or more up. So, I mean, it's not just about, counting the parameters of a theory, right?
Yeah,
I just
I
want
agree.
to see whether you agree that one should understand these things in this grander framework than just the data fitting of in a model.
I agree, but probably this love that we have for unifying theories comes again from plausibility, because with the theories that we have found so far, yeah, this is a huge conversation and it's very long, so we should cut here. But just briefly, I would say that I'm getting more skeptical about physical theories nowadays as describing nature rather than describing the way in which we interrogate nature. So maybe this idea of unifying theories is just a mathematical artifact that is somehow summarizing the way in which we extract information from experiments. I'm not talking just about data fitting. This is the easy way. That's why I'm doing that, because I'm not that smart. So we're going from models to theories and all that problem. But to me, the problem is almost the same. So we know that there are symmetries in nature because we have measured those symmetries. And now we have mathematical theories according to that. Okay, I don't know. I don't have the answer, but that's a huge conversation. So
Thank you.
not here.
So next talk is by Eduardo Bataner, full professor in astrophysics at this university at Granada. Now he's emeritus professor and has done some very recent work on the possible theory of origin of life in the universe.
Bueno, para pasarse. First of all, thank you very much to the organizers, mainly to the head organizer, Joaquin, well, I am an astrophysicist, but now I present a paper about life. But life is also a cosmological problem. Even I would say that the existence of life is the most important cosmological problem. In the universe, there are galaxies, there are stars, and they are also living beings. We could say that living beings are like black holes of complexity. Uh, if we understand Darwinism, the combined effect of replication plus random mutations and natural selection, the question is if Darwinism is able to explain to the very high complexity of the living beings. Our own answer is no, but there are many other scientists that are looking for a fourth thermodynamics law, or in other words, looking for a missing law. I reproduce here a citation of Stuart Kaufman. The funny thing about evolution is that everyone thinks they understand it. I don't, of course. Or this sentence from Goldenfeld and Wessel: the process of evolution itself, writing off as a solved problem under the catchphrase natural selection. Okay, I will present what we could call a C theory. With this theory for the complexitation in the universe. Our interest is not in complex systems themselves, but in the process of complexitation. It is important to say that this is not an alternative to Darwinism, but rather this is a complement to this Darwinism. Well, in the C theory, we postulate the existence of a C fluid with a C density, a C velocity, and a C flux, defined in all the spatial time coordinates everywhere at any time in the universe. The C density constitutes the C field. This C field is not material, but it is physical. The C fluid equations define the nature of this fluid, namely two basic equations in fluid dynamics, as are the continuity equation and the motion equation. This is the motion equation in which we see that a C flux, is generated, but the gradient of complexity, if we understand complexity as 1 over the specific entropy. Due to this fact, the C fluid tends to be concentrated in low entropy systems, as are living beings. And the other one is the continuity equation, which is the C mass conservation. Here it is written in a very common and well-used continuity equation. And if we integrate it in time, that means that the C mass in a system, well, the C mass of the whole universe is a constant in time. Uh, so this is the what the sea field is. Now we will see what the sea field does. The distribution of mutations with entropy should be non-symmetrical. Mutations with a decrease of entropy should be favored. Therefore, we assume the existence of C mutations which, which being the standard random mutations being not excluded. And these C mutations are induced by the C density. C mutations are also random, but with the ligature delta S being S the entropy less than 0. And we make a hypothesis that the increase, in this case the decrease of entropy, due exclusively to the C mutations is proportional to the C density. And this is the evolution of a species if only C mutations were at work. I must say that natural selection is always present, but natural selection is not in this C model what drives evolution. At least it is not the only driver, but it is always the judge deciding which new species can survive. And with the above equations, we can obtain the evolution of the specific entropy of a species as a function of time. Of course, considering the isolated effect of C mutations. If we plot this equation, we see here this plot. So here is entropy, and in the opposite direction we have the complexity along the time. We see that at the beginning the C mutations have a low effect to decrease the entropy because the slope of the curve here is very low. But at the other end, this other place, when there is also already a large complexity, then here the effect of decrease the entropy of the system of the living being is much more effective. This could be explained this complexity breeds complexity. So C and standard mutations will act together, but at the beginning, where the C mutations are not so important, for instance, for bacteria that are not very complex, sorry, I know that bacteria are very complex, but not so much, standard mutations would dominate, and interesting models like this one by Silexi and Muñoz, for example, and the reference theory should be predominate. In these models, the adaptation predominates. For more complex animals, C mutations become more important, and the result is adaptation plus complexitation. So there is a feedback mechanism then, because here we have a big complexity, 1 over S. This is the, it produces in a increase of C density. And the C density, as we have seen, produce a decrease of entropy. Only in this feedback, in this loop, there is a very fast branch. Here, this is in fractions of less than 1 second. And the other hand, and the other branch takes place in typical evolutionary times, like kilo years or so. Well, with the above equations, with a typical decomposition of the sea density, for instance, a mean value and a modified small value due to a perturbation, and then we obtain very easily an equation of waves. There are waves. Well, this is not surprising because in fluids there are waves. It is the sound, and here we obtain also an equation of waves. That means that the waves exist. These sea waves, and the speed would be of the order of the velocity of light, or at least very high. Under the wave-particle duality interpretation, we could speak about another particle, which is the viton. How to produce sea waves? For instance, a sudden change in entropy generates a sudden change in density, and the sea waves is generated. A sudden change in entropy may be a simple mutation. There is an important simile that explains better what I am saying. The energy, the particle is the photon. And on the other hand, this simile introduced the viton as a particle for the entropy. So the viton could be a particle of information. Here we find several results that have some relation with the viton. For instance, Vlatko-Vedral, we should no longer think of the most elementary units of reality as fragments of energy or matter, but we should think of them as units of information. Or this quotation from Melvin Gibson, information may be stored in elementary particles just as DNA stores information. This information might have mass, implying a physical presence. Have viton mass? Well, I don't know. If yes, what mass? A viton could tell a DNA receptor the delta S value of the mutation, but it could also provide the energy to carry it out. In this case, the mass of the viton could be the binding energy involved in the mutation. This is of the order of 0.1 electron volts, lighter than neutrinos. So very difficult to find from an experiment point of view. Well, let us speak about the possible advantage of the sea theory. It is presented, as I have said, as a complement of Darwin theory. It makes evolutionary processes more efficient and faster, and therefore more credible. Darwinism is only demonstrated for microorganisms, and in this case, the sea theory provides similar results than Darwinism. The origin and the evolution of life must be considered as a unique problem because usually they are considered two different problems. Because if mutations are the what drives the evolution, and of course before the uh, establishment of the DNA code, there was no. So I mean that we must consider origin and evolution as parts of the same problem, maybe with different mechanisms, but arising from the same principles. For instance, gold uh, speak about a world. Before the world, the world is at the beginning of a species uh, explained by Darwinism. But what is the problem before the world? There is always spoken about a soap, but well, I don't like soaps. Monod considered that before the war, this was a monstrous coincidence, therefore life would be exceptional. Uh, so both problems, origin and evolution, should obey the same principle, although with different mechanisms. Uh, After the gold world, once the genetic code is established, entropy changes are due to random plus C mutations. But before the world, uh, thermodynamic fluctuations in an equilibrium system involve specific entropy fluctuation. Due to fluctuation, there are fluctuations of the real density, the pressure, the temperature, and also the entropy. And if this entropy fluctuation cause a seed density fluctuation in turn, which cause the decrease of a specific entropy, albeit over a larger time. Uh, another uh, Another concept is the what we could call enally, the growth of entropy inhomogeneities is favored with this equation, with this C theory, uh, because here we find that contrast animals and interanimal medium. In other words, we have a cat. Here there is no cat. There is an inhomogeneity in the growth of entropy. For instance, there are many others. Enalic examples. For instance, there are stars and interstellar medium. There are galaxies and intergalactic medium. There is a large-scale structures of the universe with large filaments and large voids. Here we have a picture of the universe as observed by the cosmic microwave background, and we see a complete isotropy. But time, there is an evolution, and today this is a picture that reproduces the complicated distribution of matter with large voids and large filaments. Some thermodynamics thoughts. The first law is the energy is conserved. The second one is the entropy does not decrease. And the fourth could be the enally does not decrease. These properties, of course, only in isolated systems. The universe maybe is an isolated system. The energy of the universe does not decrease. This could be an arrow, another arrow of time. Other examples of energy, the population. There are large cities, towns, small villages. For the wealth, there are millionaires and poor, because they obey very similar equations. Well, uh, about the possible advantages of sea waves and vitals. Uh, Mutations occur first in individuals. How to extend the mutation of an individual into mutation of the whole species? Darwinism explained that, but requires a lot of time, much simpler and faster with sea waves. The mutation of an individual DNA produces delta S, hence an increment of sea density, then emits a sea wave with delta S, which can be absorbed, and so on and so on. And therefore, um, this is much more easier with vitals to solve this problem, which is a problem in any case. And that must be the case of co-evolution. Panspermia. Panspermia is a well-known theory that assumes that the origin of life is extraterrestrial. Sea waves can propagate in vacuum, and it is better to transmit delta S, the decrement of entropy, by vitons than through spores. So Viton-induced panspermia is much more believable. This could introduce new light for the problem of the very rapid appearance of the first microbes in less than 0.2 gigayear. About this complexity breeds complexity, could be provide some new light about the rapid cerebralization of hominids. Darwinism, with random mutations, introduced adaptation. The sea theory, with random and sea mutations, introduced adaptation plus complexitation. Adaptation and complexification are not synonymous. They may have some relation, but they are not synonymous. So, well, what is the adaptative advantage of a Beethoven symphony? And about the problem of horizontal gene transfer, HGT. I follow here the interesting paper by Goldenfeld and Reset. Microbes strongly interact. Microbes are able to exchange genes. This is the HGT problem. How to produce that? Usually it is said that with plasmids or with transposons, conjugative transposons. Well, to emphasize the importance of this HGT, Rese says that this is the principal driving force in early cellular evolution, then this is an evolutionary force to be reckoned with, comparable in power and consequence to classical vertical evolution mechanism. Here is a well-known picture of a gene transfer from a bacteria to another with this channel of communication which is called pilum, in plural pili. Uh, in this way, there is some mechanical transfer of information between a bacteria and another. But it is necessary to communicate not with only one cell, but with the whole strain. And communication through pili is much more difficult than through sea waves. Goldenfeld and Besse says that the worldwide spread of antibiotic resistance genes across distantly related bacteria, crossing species and phylum boundaries and physical locations. I put in red physical location because if the communication is taking place in different physical locations, and then a mechanical means of transfer is impossible. And Saliers and Amabile Cuevas also say that there are still other types of conjugal gene transfer that have yet to be identified. Uh, well, at the beginning, this horizontal gene transfer was considered an evolutionary curiosity and now is considered a major force in prokaryotes that bacteria and archaea that communicate quickly and effectively. But even this is in eukaryotes, both microbes and human cells. They communicate for to communicate the antibiotic resistance gene. Uh, so sea waves would be the most effective way to cell-to-cell communications, important in evolution, macroevolution. Well, many thanks.
Thank you, Eduardo. Thanks a lot. Any question?
Thank you. It's interesting talk. So I have some problem with these sea waves. So you said that these waves exist, they will transmit very fast, no? But you need some medium. For example, I understand it's matter. You said it's matter. No? And that means that in order to have an effect over a species and produce a mutation, must be species in this space, no? in this place. No? So then these sea waves cannot transmit from one part of the universe to another part of the universe that is far away, because there is no medium matter, well, it's empty space more or less. No? So I think you need a kind of excitable system, excitable medium for this type of wave to have an effect, if it exists, and induce a mutation, including this thing.
Well, the medium of these sea waves may be the vacuum. So this is, for instance, like the medium is on medium. This is the vacuum. Is here also the medium could be vacuum. So I can consider that. I have
So any other question? There. Some.
Thank you. That was a very interesting idea. At the moment, we're seeing artificial intelligence becoming rapidly more complex. So, in your theory, do you think this mechanism plays some role also in artificial systems? So, could the evolution of AI be accelerated by this kind of thing? I'll say it louder. So, artificial intelligence, inteligencia artificial, could this mechanism play a role in the development of artificial intelligence getting more intelligent, more complex somehow?
With your Python or whatever pieces of information for months and also interact with topics.
Um, yes, we have introduced some new light about the cerebralization of humans, and therefore this Python could produce decrements of entropy, which means to increase the degree of complexity. Therefore, if this is so, we could expect that the intelligence of the future humans would be much more, much higher.
Thank you very much. So I think it's time to close the session, and thanks the speaker again. But before we're stopping here, we don't have any more talks, but there are a couple of announcements by the organizer.
Thanks a lot for attending. So tomorrow we are planning to take the official picture of the Granada Seminar. So we thought that the best time to do is after coffee break. Okay, so I think the good site to do is down in the coffee break because this nice building in which we are celebrating the Granada Seminar. So it is a good place to take the pictures. Okay. And the second thing is for a speaker for tomorrow. So please send me your PDF of your talk in order to avoid any technical issues with the presentation. If you think you can do with your own laptop, you know now what are connections. So, but please try before in order that control that everything is working properly. Okay. So that's it. So thank you.